some great speakers here, and we're going to get started with Eliza Harrison. And the title of her talk is a seaweed cultivation demonstration project led by Ocean Rainforest. All right, take it away.
Thank you. I'm going to move into blinding light, I think is where I'm supposed to be. Anyway, well, thank you so much. It's such a pleasure to be here. My name is Eliza Harrison. I work as the program manager for Ocean Rainforest. And today, I'm going to be giving a little bit of background on an offshore seaweed cultivation demonstration project we have been running in the Santa Barbara Channel for about a year now, and will continue to run through 2023. But before jumping too much into the project, I thought it would be helpful to give a little bit of background on who we are as a company. And essentially, Ocean Rainforest was founded in 2010 with a mission to improve people's well-being by cultivating seaweed in a way that also supports the health of our blue planet. We have an incredible team to do that with. We are a collection of academics, scientists, researchers, and entrepreneurs. And over the course of the last 11 years or so, have built this incredible skill set that has allowed us to begin to explore the feasibility of offshore seaweed cultivation, both in the Faroe Islands, which is where the parent company is based, as well as in Santa Barbara, California. And our work in Santa Barbara comes largely in response to a demonstration project that is funded, or in the context of a demonstration project that is funded by the Advanced Research Projects Agency Energy, which is a which is a branch of the Department of Energy. And that project falls under the macrosystems heading and falls under the macrosystems project name under the Mariner program. And so that project, is a little bit of context, is really looking at demonstrating the feasibility and testing the feasibility of growing seaweed offshore, ideally for animal feed and biofuel. So we're thinking kind of larger scale, sustainable, economically feasible cultivation of seaweed. And for the purposes of the macrosystems project, we're working with giant kelp, which many of you will be familiar with as the species of kelp that is growing off the coast right now. Beautiful long stalks of seaweed and plus with blades, sometimes a nuisance for surfers, but an incredibly valuable and regenerative resource, which is why we're interested in growing it. The purpose of the Augyte project, at least from our perspective more specifically, is to demonstrate or test the feasibility of a cultivation system we've designed to ensure that it's scalable and sustainable in terms of its operation, predictable in terms of how it behaves in the environment, profitable because that is ultimately how any industry really gets off the ground, and survivable, so be able to withstand offshore conditions in Santa Barbara Channel, as well as in other parts of the California coast. And phase one, there were two phases of the Augyte Mariner program. Phase one was really focused on the modeling aspect. So understanding what the techno-economics would be. So conducting a techno-economic assessment, running a finite element analysis on the structure we've designed to ensure that the system, which really is in essence comprised of ropes, buoys, anchors, mooring lines, these fairly kind of common devices that are used in the marine environment, to make sure that that system is able to withstand those offshore conditions. So a lot of our work in phase one, which ran for about a year between 2018 and 2019, was looking at the modeling side of things. And then in 2020, our team, Ocean Rainforest as the prime contractor, was awarded this phase two grant, which gives us, or contract, excuse me, which gives us the opportunity to test this system in a real world environment. So what we're looking to do, and have tried to do since 2020, since the summer of 2020, is to de-risk all aspects of the seaweed cultivation value chain. So that goes from seeding to ultimately harvesting the biomass. It's also looking at demonstrating the capacity and the capabilities of that cultivation system, specifically in terms of how much yield we could get from the system at any given point in time. And that will help inform the economics of our work. And the last piece is to optimize different aspects of cultivating seaweed so that collectively we can begin to move these aquaculture practices into an increasingly sustainable, state-of-the-art way of interacting with the marine environment. In the context also of the Augyte program, we have a collection of experiments that need to be executed really all in an effort to inform the amount of seaweed that we could potentially grow. And many of these are related to novel seeding techniques. A number of them are also related to different harvesting strategies. And then of course, different ways of utilizing ocean space. And that is what kind of the grow line spacing and grow line length are looking at, is how do we maximize the area of the ocean that we've got to grow as much seaweed as possible. Unfortunately, in partnership with a number of excellent local collaborators in the area, we've been able to already test some of these experiments. So over the course of the first year, we have installed a hatchery facility in collaboration with the Cultured Abalone Farm. Doug Bush is here today, so he's an excellent resource if you're interested in learning more about that. We've also been able to propagate and induce biomass that's all coming from kelp beds in the area. So all locally sourced native species of seaweed that we've then actually been able to outplant as well at an offshore cultivation site, the Santa Barbara Mariculture Farm. And in parallel to our direct work, working in growing effectively what are considered seaweed seeds or gametophytes, we've also been able to test a different seeding technique. So that is a technique called direct seeding, which simply is less labor intensive than many of the traditional ways of seeding seaweed on open ocean farms. And the benefits of direct seeding are that it requires less time in the hatchery. So you have fewer resources you use, you have less energy that's being used, you have less time that's required for people to work and care for these gametophytes. So it's an incredible advancement for the industry as a whole. And what we did over the course of the last year and will continue to do going forward is to demonstrate that direct seeding actually works with giant kelp. And as far as we know, at least in terms of the literature review, ocean rainforest deployment last year of direct seeded kelp lines was the first successful use of direct seeding technology, which has been used in other species of seaweed, but it was the first successful use for giant kelp. So we as a team were quite excited about that milestone achievement. And you can see here a few pictures of the sporophytes growing on our ropes in the lab. We've also been able to work, as I said, a little bit with the Santa Barbara Mariculture group and Bernard Friedman to install 6 lines at their project location to then deploy and test the feasibility of that cultivation system that we've proposed in a small scale manner. And so what we've done at the site is modify the existing infrastructure on his lease to facilitate and to allow for our cultivation system to be installed there as well, so that we can begin to understand how it behaves in the environment. We've also implemented a bi-monthly monitoring plan. And the purpose of that is to really understand the environmental parameters that inform and can support seaweed growth, but it's also to understand what the potential impacts of seaweed cultivation might be in this area, because ultimately we don't want to have any sort of negative impact. We want to understand how the natural marine environment interacts with our seaweed cultivation system and then minimize any sort of adverse impact. And just as a little bit of a time frame, you can see here over on the left-hand side of the screen, the lines that we deployed in March 30th of this year. Pretty bare looking lines, very empty. And then on the right, you see a picture that was taken on August 16th, so about 2 months ago now, of our cultivation system in the water full of seaweed. And if you ever dove a kelp forest, to me, that cultivation system looks a lot like a natural kelp bed. And that's what we're trying to do. We are trying to create a system that behaves like a natural rainforest, a rainforest in the ocean, if you will. So going forward, years 2 and 3, we'll continue our work at the hatchery facility, iterating on different seeding techniques. We'll also continue to fulfill a lot of those other experiments in relationship to harvesting and optimizing our cultivation system. And then we'll also continue our monitoring plan, because again, we've been out in the water for a year. We have 2 more years. That will give us a lot of information, a lot of data of how the environment interacts with our seaweed cultivation facility. And then in parallel to all of this actual work in the hatchery and offshore, Ocean Rainforest submitted a permit application to the U.S. Army Corps of Engineers for a demonstration project about 4.4 nautical miles off the coast of Santa Barbara. That application was submitted in September 2020 after a lot of stakeholder engagement, and we have since, I'm very excited to share, secured approval from the Department of Defense, the U.S. Coast Guard, the California Department of Fish and Wildlife, and as of last Friday, the California Coastal Commission. So we are teed up to really begin to execute what would be, again, a demonstration project, a little bit bigger scale, further offshore, but to again understand how this industry can be built in a way that's as regenerative and sustainable as possible. Which brings me a little bit to why we're interested in doing the demonstration project, which is essentially to test whether or not we can grow seaweed at a scale in a sustainable manner. Another piece of our work is improving and advancing state-of-the-art aquaculture practices in relationship to seaweed cultivation. There's also, as we're all here today, a community education and engagement aspect, which is ultimately how the industry will continue to grow and develop. And is also promoting the supply of sustainable and safely sourced seafood, both in the US and internationally, but particularly in California, because that will really set the stage for a new relationship that we would have with locally produced seafood. And the reason perhaps that this is most interesting, at least to me, as well as interesting to all of you, hopefully, is that seaweed is an incredibly regenerative resource. It carries a number of environmental benefits and actually addresses many of the UN Sustainable Development Goals that have been generated over the course of the last few years. A few of those to highlight in particular are the fact that seaweed cultivation, as you saw earlier with that picture that looks like a kelp, natural kelp bed, it creates ecosystems, so it can provide nurseries, feeding ground, habitat for marine life. It also removes meaningful amounts of carbon dioxide and reduces ocean acidification, so addressing climate change. Another component is removing excess nutrients from the marine environment, reducing the risk of harmful algal blooms, and beginning to address some of the excess nutrient pollution that has proven such an issue in many parts of the United States. And lastly, last but not least, it also provides a unique economic development opportunity and supports economic diversification in coastal communities by creating these very blue and sustainable jobs. And with that, I would like to thank you all for listening. My contact information is on the screen. If you have any questions, contacts, comments, concerns, definitely let me know. I'm always happy to help. Thank you.
So we'll have Amanda set up her presentation, and we'll see if anybody has any quick questions from the audience. Are there any questions? Yes.
I'm not sure, I might have missed if you addressed this, but what's the timeline or projected timeline for that offshore farm you mentioned towards the end?
Yeah, that's a great question. So we have been very fortunate to work with a number of regulatory agencies. The timeline for that is by mid-December of this year. We expect to understand from the Army Corps whether or not we will have secured the permit. And then the permit will run for 2 years, really again, to demonstrate whether or not this is going to behave, the system is going to behave as we think it will. So that's the timeline.
Yeah, question back there.
Is the grant that the U.S. Department of Energy funding that you guys got covering your monitoring costs, or is that privately funded through the company?
Yes, so the contract we have with the U.S. Department of Energy is also to address monitoring, because of course they want to understand comprehensively what it takes to cultivate seaweed at scale, and monitoring is a critical piece to that. That will continue though as we move forward in the project.
Question right here.
Yeah, can you talk about the physical structure of how the farm is set up? I saw you had a diagram of kind of a diagram, like the acres set up, but could you just give us an explanation of how it's physically built?
Yeah, of course. So the system, as I said, is kind of made up ropes, buoys, and anchors. You've got a collection of backbone lines that almost form a square. And then within that system, you've got cultivation lines. And so those will run along the short side of the rectangle. And from those cultivation lines, you'll have grow lines. Those grow lines are ultimately what's seeded with the seaweed seeds, and is intended to maximize the use of ocean space, because you're able to kind of dramatically increase the amount of area that you have if you're adding additional line, but in a vertical sense. And then that system is secured in the environment and kept taut by these long mooring lines that go down to a collection of anchors that are placed in sandy bottom habitat. Does that help?
Yeah.
All right, I think we have to move on for the sake of time, but thank you again, Eliza. That was fantastic. Thanks, everybody that just came in. So our second speaker is Amanda Swinimer, and she's going to talk about seaweed, ocean's healer, how seaweed nourishes and cleanses both the planet and people. Take it away.
Thank you, and thank you to Simona and all the organizers for this wonderful event. I'm really excited to be here. I came all the way from Vancouver Island, and yeah, I'm really, I've been harvesting edible seaweeds for 20 years. I have my own business there, but I've gotten really interested in seaweed and health, and I want to talk today about the parallels between how seaweed helps to heal both our environment and how it's connected to also healing ourselves. Well, seaweed is an amazing purifier. It absorbs toxins in the ocean, and incredibly, it absorbs the inorganic form of heavy metals. And because that form of the heavy metals is toxic to seaweeds in the same way it's toxic to our bodies, the seaweed transforms that molecule into an organic molecule, which is essentially biologically inert. Seaweeds also are an amazing purifier in our bodies, and sodium alginate is the absolute hero of purifying toxins from human bodies. Sodium alginate is located in brown seaweeds, and it binds in the gut. It binds to certain toxins, including certain heavy metals, dioxins, PCBs. It forms an insoluble salt, so it's not going to be dissolved in the bloodstream, and then it's safely removed through the stool. Seaweed is also an incredible nourisher. And I say seaweed, I'm including all the algae, the microalgae as well. So the algae are the primary producers of our ocean. That means they are the foundational nourishment for everything from the tiny zooplankton all the way up to the mighty orca as the apex predator. They are providing this nourishment for the largest ecosystem on Earth, the ocean. And kelp forests, which grow on almost every temperate coastline and are in a global state of decline at about 2% per year, are one of the most productive ecosystems on the planet. Seaweeds are also incredibly nourishing to humans as well. They are the most concentrated food source of minerals on the planet. They're rich in vitamins, protein, omega-3s and essential fatty acids, and they're often the 3 to 6 ratio of omegas is often in a 1-to-1 ratio, which is really ideal for human health. And also it's been recently discovered that they're a great source of prebiotics. So they're literally nourishing the microbiome of our gut, which we now know is responsible for the large part of our immune system, for brain health, and overall human health. Seaweed as protector. Again, I'm talking about algae as well here. So it produces an estimated 50%, with some estimates as high as 80% of global oxygen, absorbs an estimated 1/3 of atmospheric carbon dioxide. And when microalgae die, they release dimethyl sulfide, which reacts with oxygen in the atmosphere to create aerosol sulfites, which act as nuclei for clouds. And that process is critical to cloud formation and the regulation of global climate. And seaweeds also help lower the acidity of the ocean, which is increasing due to climate change. So it can have a real acute impact on species such as shellfish, where seaweed are growing. Seaweed is also an amazing protector in humans as well. So I'm just going to briefly mention 3 of the top unique compounds to algae that really impact human health. The first one is fucoidin, which is located in the cell walls of brown seaweeds. And fucoidan is the subject of well over 1,000 peer-reviewed published studies. It has multiple mechanisms that in preventing and fighting different types of cancers, and this research is mostly on human cancer cell lines in vitro and animal models in vivo. Also just a range, immune enhancing, and a whole range of protecting the body's systems from harm and keeping them healthy. The second molecule compound that I wanted to mention is fucoxanthin. It's also in the brown seaweeds. Fucoxanthin is used to help harvest light for photosynthesis, and it gives the brown seaweeds their golden hue. Fucoxanthin is also the subject of many studies. It has also shown several mechanisms in preventing key processes of cancer, as well as preventing other chronic diseases such as diabetes and cardiovascular disease. And the last compound that I want to talk about is a group of compounds called fluorotannins. And this is a group of phenolic compounds, and they are in all three types of seaweeds, the reds, the greens, and the browns, but they're most abundant in the brown seaweeds. And the molecule I have here is a particular fluorotannin called diethyl, and it's extracted from Aclonia cava, and it's considered one of the most powerful antioxidants known on Earth. And as a group, fluorotannins are one of the most studied marine compounds in terms of human health. And again, a whole range of protection. In the cell, in the algae, the fluorotannin provides structure to the cell wall, helps protect the cell from UV radiation, and it serves other biological functions. Seaweed and resilience. So seaweeds grow in the most extreme environments. They're subjected to wild variations in temperature, like twice daily whenever the tide's going in and out. They're alternating between air and water. Sometimes they're freezing and thawing twice within a 24-hour cycle. And they not only survive, but they thrive. If you've ever been to a rocky shore, you've seen how well the seaweeds do there. So the adaptations to these extreme living conditions have resulted in a medley of novel therapeutic substances that are amazing for human health. There's evidence that they prevent protein misfolding. There's a study going on a seaweed called Alaria esculenta that grows in the Bay of Fundy, which is one of the most extreme conditions. It's exposed to freezing and thawing and has to prevent its own proteins from misfolding so it can survive. And it's shown to prevent protein misfolding in amyloids, so misfolded proteins associated with Parkinson's disease. Also developed hundreds of unique antibacterial properties. Seaweeds are exposed to airborne and marine bacteria, and as well as hundreds of unique antiviral activities, including antiviral activity against herpes, HIV, and HPV. Seaweed adds beauty to the ocean. If anyone's seen seaweed growing in its wild habitat, I need not say anymore. Here we have the absolutely beautiful iridescent Mazaella splendens, or rainbow seaweed, just wonderful. And I think that the beauty of seaweeds definitely needs to be mentioned. It provides a lot of beauty to the ocean. It also helps humans be beautiful. Seaweed has been considered for probably millennia to be the fountain of youth. It's used in all kinds of creams and things like that to reduce the signs of aging. It's been used for many generations to enhance one's natural beauty, to enhance one's overall vitality, to decrease stress and induce calm, and specifically to nourish and tone the skin and the hair. So I just wanted to say thank you again to everyone. I'm thrilled to be here. I invite you all as well at 2 p.m. I'm going to be speaking with some other amazing folks on a panel discussing seaweed and health. at the outdoor tent. And thank you again to the organizers, and thank you for being here. And if there's any questions, please let me know.
Are there any questions for Amanda?
Yeah, go ahead. Could you say again the name of the seaweed that was studied with the prevention of misfolding protein?
Yes, Alaria esculenta. So you would have Alaria marginata, maybe not in Santa Barbara, but going up north, you would have Alaria marginata as a similar species.
Thank you.
Yeah, question back there?
Yeah, you talked about fucoid down. Is that found only in the fucoids or is it in all ground?
The fucoided? Sorry, you're right, no, like,
The fucodan.
fucoxanthin.
Yes.
Yes, it's unique to brown seaweeds.
But not just the fucoid seaweeds.
No, it's in all brown seaweeds that I know of that have been tested, because it's what gives the brown seaweeds their color. It's part of their. It's not directly involved in photosynthesis, but it's involved with the light harvesting for photosynthesis.
Last question.
You described seaweed as being near miraculous as far as therapeutic powers. I should be at the beach eating it daily.
But you should be, but the great thing about seaweed, it's so concentrated in these nutrients that you don't need to eat a lot of it, like as little as 5 to 8 grams a day, which is like a teaspoon of the ground up seaweed, is really all you need to get the therapeutic benefits. Like you want to be consistent about eating it daily rather than eating a whole bunch of it every once in a while. But yes, I agree with that. Thank you very much.
Okay, so next up we have Erika Thalman, and she's going to let us know a little bit more about culturing red seaweed at HSU. So this is my alumni too. I'm proud HSU graduate, so thank you so much. I want to hear more.
Hello, everyone. My name is Erika Thalman, and this is my first Seaweed Festival. I'm very excited to be here and to be with all of you and to share my project with you. My thesis is on Pacific dulse and growing it in Humboldt Bay on an open water farm. Next slide. So a little bit about seaweed farming. It's not a new practice. It is ancient, going back to about roughly 500 years. However, today it is considered a relatively new industry, particularly in the US. Next slide. I don't have to explain this to you guys, as you are all seaweed nerds. Why seaweed? It's in everything. It's a very popular ingredient, incredibly useful, and as we just heard, extremely nutritious and valuable in so many aspects. And because of its usefulness, it has an increased global interest, particularly growing it as a crop. Next slide. About 97% of the world's seaweed comes from farming. However, the United States only adds about 1% of that. We import most of our seaweed, costing us about $50 million a year. And California, specifically, we rely mostly on wildly harvested seaweed. And while right now that is, it is done sustainably with the growing interest and the market rising for it, farming will have to begin contributing to that in order to keep seaweed sustainable. However, California does have very strict regulations. While seaweed farming is growing in the US, it's primarily on the East Coast and in Alaska. And the restrictions here have been hindering the growth of it in California. Next slide. However, the waters began to change in 2020 when HSU partnered with Greenway, built the first commercially licensed seaweed farm in California state marine waters. How this farm is different from a lot of others, It's in open water, it's not land-based, so you don't need land, you don't need buildings, you don't need fertilizer or electricity to grow your seaweed. While there are, I think, 1 or 2 other open water seaweed farms in California, they're still in the research pilot phase and not quite there at the commercially licensed place, but they are getting there. And I strongly encourage that. I hope more farms get in. But being the first in a lot of ways, particularly being the first in Humboldt Bay, we bear the burden of being everyone's guinea pig. There's a lot of potential farmers that want to begin seaweed farming there because the Harbor District went through the process and has pre-permitted sites available for shellfish and seaweed culture. And many of the shellfish farmers also want to add additional crops since they have seaweed growing on their gear anyway. So a lot of the potential farmers are looking to us to see what they need to do, what they probably shouldn't do. And we're also working with regulating agencies who are still in the process of learning how to regulate seaweed because they haven't really done it before until lately when a lot of seaweed farms, land-based and our open water, have started to pop up. Next slide. So my specific research focuses on Pacific dulse and whether or not we can even grow it in Humboldt Bay. And if it does, which it does, we wanted to find the optimal growing conditions. I was specifically focusing on growing depth and season. I also wanted to look at what nutrients this seaweed could pull from the water, as it has the ability to absorb nutrients, particularly carbon. And I wanted to test for heavy metals and pesticides because our pre-permitted site is next to an industrial area, and it also has an old shut down pulp mill, which has had a leak in the past. So we want to make sure that our dulse, which is primarily used as food, is actually safe to eat. Next slide. So my farm design consists of 2 long lines. It has a main surface line attached to 2 mooring buoys, which are attached to 100-pound Danforth anchors to anchor ourselves into a muddy bottom. And off of this, we have what we term droppers. This is a shortened version of what I have on my farm. It's just sort of like a little attachment loop. And then we have our seaweed at 0, 1, 2, and 3 meters. So that's the way we can figure out what depths work best. And we have them spaced out every 2 meters to account for COVID testing. We originally wanted to do every 1, but COVID was actually a blessing in disguise because it's nice only having to work with 100 of these instead of 200. And that picture is where we were actually dropping them in the water. It was such an exciting day. It was postponed and postponed, and finally it happened. Next slide. This is a picture of the actual farm in the water. You can see our long lines with this colorful parade of crab buoys. And then surrounding it, we have 6 orange and white navigational buoys to warn motorists that there are submerged lines, because it can be really hard to see, particularly if it's rainy or foggy. Next slide. So I've been asked a lot, why Pacific dulse? Like, why red? Why not do something more charismatic like a kelp? Well, our main reason why is our pre-permitted lease only allows us to do a handful of red seaweeds, including Pacific dulse. We also chose it because it's a native and locally found, making it easy to collect, and it promotes the growth of native seaweeds. 3, there's a ton of information available to help us grow it. While not a lot of research has been done specifically on Pacific dulse, its cousin Atlantic dulse has been a prominent research subject in aquaculture for quite some time. And because of their similar life histories, we can draw from Atlantic dulse to help us design and carry out our project. And the 4th reason is because we can grow it vegetatively. We don't have to use spores in order to seed our lines. Dulse is one of those seaweeds where if it is detached from its holdfast, it will be able to survive as a dripped algae in the water. So we took advantage of that And we collected it locally, about 30 minutes up the road in Trinidad, California, from our farm site. And then we fragmented it, and we put it in what we call an integrated multi-trophic system, which is that little system over there. We paired it with some sablefish. The sablefish made the nutrients that helped the seaweed grow. And then the seaweed took out the nutrients so that the fish had more suitable water to live in. So luckily, the IMT was also a blessing in disguise because COVID delayed our deployment of our lines. So we needed somewhere to keep our seaweed, and this allowed us to grow excess. So when we seeded, we had some extra left over, and we can use that extra to continue to seed our farm. We don't have to draw from wild populations anymore. Next up, oh, give me back, sorry. Seeding the actual dropper lines. So since we don't have to seed them with spores, what we decided to do is we had these twisted nylon ropes. All we did was sort of untwist it slightly. We inserted a 15-gram bundle or a frond of dulse, and we closed it. The twist naturally can hold that dulse in place while it is in the water. Next slide. This is us actually deploying them. It's not as simple as it looks. I mean, it is simple in theory. We just sort of like, we were attaching them with bait pins. We probably should have tied them, but Raphael, who's sitting right here, and Karen, bless their souls, were the ones in charge of getting those bait pins through the rope. It wasn't easy. It was a very, very strong twisted rope. Next slide. So results, finally the big day. We pulled about 38 pounds each from our 2 trials. My first trial was from September to December in 2020, and then my trial 2 was from April to July. Longline 1 did go in 2 weeks prior. Longline 2 had to go in a little afterwards due to some technical difficulties. So I split them up to show you the difference. We did have a growing window earlier on in the season that we did miss with our Longline 2, which I thought was important. Next slide. And then I want to show you some cool things that I learned as a seaweed farmer. So one thing is that dulse is a shape-shifting seaweed. Here was one of the things that we had planted earlier on. It has very, it's like, you can see it's bushy, very tiny little blades. However, with trial 1 going on into late fall and early winter, the blades started to get very flat, very clean, very wide, and having a lot of growth proliferations on the edge. Next slide. Also, that was also reflected in early spring as well. And this I wanted to show you guys because it shows the true potential that Pacific dulse has as a farm species. These are both my 0 meters, my hand for scale. That came from a 15-gram bundle like that. And that's all the seaweed that you got after that. Had I loaded my lines, I would have had way too much seaweed to deal with. Next slide. The biggest problem that we had in Humboldt Bay in particular was biofouling. Trial 1, we had more of these invertebrate hydrozoans that you can see here. Trial 2, we had some hydrozoans, but mostly barnacles. And the biofouling is worse when the water is warm. So, which you're going to see once June hit, that's when the biofouling got bad and the seaweed basically stopped growing. Next slide. And number 4, it's an unpredictable and very hard job to do. On average, we spend about 4 hours on our stomachs, pulling a boat along, looking up seaweed and stuff. Sometimes a storm will come through and send a gigantic island of bulk kelp through your lines or a log. And sometimes if it's raining, you still have to go out. If it's windy or the current's too strong, you have to cut your trip short. But all in all, it was a learning experience that you could only get from actually doing it. Which is, I'm very, say, blessed, but also kind of cursed. Next slide. Okay, so we harvested. What next? While we are commercially licensed, we decided to donate our harvest to a local farmer who's actually here, by the way. If you see Mika, recommend talking to him. But yeah, so Mika made a bunch of compost with her dolls and gave me a bouquet of beautiful flowers, and it was gorgeous. Dolls compost works. Next slide. Okay, so I realize I'm running low on time, so I'm going to breeze through this really fast. For trial 1, both 0 and 1 meter did relatively good as we got into late fall and early winter. 2 and 3 meters sort of took a dive. Next slide. Going on into trial 2, we had a lot of growth early on. It was just like, it was impressive. It was only in for a few weeks and it was like, bam, giant bundles everywhere. And we're just like, oh my gosh. Yeah, so, but then day 86 or June, that's when the biofouling got instantly bad and the seaweed didn't really have a good time recovering. Next slide. You can see how that same trend is same for longline 2. However, at a very reduced growth, we missed that upwelling window. So it didn't get a good start and it didn't have a great finish. Next slide. This is just sort of comparing the growth all next to each other. Next slide. So loss can happen from a lot of things. Sometimes it's user error, like I'm lifting up a line and I jerk it too hard and the seaweed falls off. Or something comes through and yanks it off, or crab eats it, or the current is too strong. Trial 1, for some reason, we lost 50% from all depths. We don't know why, it just did. However, trial 2 showed a definite trend. 0 and 1 meter were the healthiest and strongest ones, therefore they had the least amount of loss. The biofouling was the worst on 2 and 3, therefore they became really weak and slumped off like nothing. Next slide. So out of all trials and like the total amount of biomass, net biomass, we're taking out the initial stuff we seeded, we extracted 18 pounds of carbon out of the water in Humboldt Bay, just with, it was like 60 or 70 something pounds worth of seaweed. Next. Heavy metals, we didn't really have any going over any dangerous thresholds. Next. Pesticides, we had no positive results, which is great. So meaning the seaweed is safer to eat. It was also interesting since it is in an industrial site, why it didn't have more. Next slide. Okay, so in conclusion, if you're growing seaweed in Humboldt Bay, particularly if it's Pacific dulls, we recommend growing it from near surface down to 1 meter depth. Late fall and early spring can be optimal outplanting times, just due to the lack of biofouling and increased growth. I recommend taking seaweed out before June. I've talked to several other farmers and they all had the same problem. Once June hits, your biofouling takes over and it's not worth anything. And number 4, your large bundles have a lot of drag, so currents ripping, it's more likely to take your crop away. So if you have a large bundle, go ahead and harvest it or trim it to reduce the size and the drag. Next slide. Just because I never got to test the one season I did really want to test, I'd recommend trying from January to March just because growth was very promising in late winter and in the spring section of my second trial. To reduce loss, maybe the use of bags or to contain your seaweed on your lines, or doing multiple harvests, trimming instead of just doing a total harvest of your seaweed to keep your sizes down. And because I only looked at vegetative growth, if someone would like to, in the future, look into spore or hatchery protocols, that would also be cool. Next slide. This project wouldn't have been possible without the CSU's generous funding through the Agricultural Research Institute, as well as HSU's sponsored programs. Next slide. And I'd like to thank you all for coming and listening to me today. If you have any questions, you can find me after, or you can send them to my email.
Thank you so much, Erika. That was really fantastic. Does anybody have any questions for the speaker? back there.
Hi, I was wondering if you could elaborate a little bit more on biofouling.
So I didn't quite hear that last part.
And like the conditions that lead to biofoul.
Okay, so conditions that lead up to it would be increased light and increased temperature, as well as increased nutrients. So as you go on into summer, you'll be getting a lot more light and warmer temperatures. So, and the biofouling tends to be species that grow really fast, so they can potentially outcompete the crop that you want to grow. So, biofouling on the seaweed, it sort of, like say for me, my crop's primary use is as food. Biofouling degrades that and it's no longer food grade, it's more like fertilizer grade. So I won't be getting, as a farmer, as high a price as I could for it. It also adds drag, makes your lines very heavy, sort of things like that.
Thanks for sharing.
There's a question right here. Do you have any possible thoughts for the remediate biofouling? Do you think it's better to avoid those high seasons altogether? If it's like a very tedious task to try to remove it?
Yeah, like a lot of times, sometimes it's like attached to your seaweed or it grows into it. So sometimes it's like very difficult to remove. For when I sent in samples for analysis, I had a toothbrush and I was brushing it. And so I would recommend, that's why I would recommend growing it in winter. While you do have reduced light and reduced temperatures and dose will grow slower, it won't have the biofouling to compete with.
All right, one quick question and then you're.
Are there weather conditions that you keep in mind? Like, for example, if a storm comes up, is there ways that you can remove the seaweed or anything? So, you know, it's not a total loss or anything like that?
So usually when a storm comes up, I just avoid going out. But I splice loops into my line, so if I do need to take my lines out, I can. But no, if a storm comes in, I just cross my fingers and hope nothing tears the farm out.
All right, thank you so much, Erika. So next up, we have Melody Jew, and she's going to tell us more about seaweed photography from cyanotypes to satellites. Take it away.
Hello, seaweed people. I'm so excited to be here today. My name is Melody Jew. I'm Associate Professor of English and Media Studies at UC Santa Barbara. And you might think, why would an English professor be interested in seaweeds? Well, why not? We're all fans here. They're beautiful and gorgeous. And I want to talk to you about photography today. So usually when we think about photography, we might think of digital photography, beautiful underwater images that are very, very clear and illuminate the kelp in fun kinds of ways. I have a lot of friends who are into this. I myself am a scuba diver. I wrote a book called Wild Media, and in this book, I try to think about how concepts are different underwater. So it's in real philosophical territory, but I also feel, I guess, grounded or floated rather by all the opportunities to be in the water with seaweeds. But the question I want to explore today with you is, in what ways do seaweeds help us take photographs of seaweeds? So I'll go into this in some hopefully interesting ways. When I first got into seaweeds, this was all because of a student of mine. I was teaching a course on ocean media, so lots of documentary film and other media forms, and my student told me that on our campus we have the largest Southern California collection of seaweed specimens at the Cheadle Center for Biodiversity and Ecological Restoration. So I started looking at these and was just amazed by how they sort of fade into the paper or look almost like plastics. There's so many people here today who are touching on exactly these things. But also at CBER, right outside the wall of the algal herbarium, were these beautiful cyanotypes, or images created by putting seaweed on photosensitive paper. So I started looking into the history of this. And it turns out that cyanotypes were first developed by William Fox Talbot in 1840, it was actually 1842, but he published a book called The Pencil of Nature. And if you think about how beautifully complex seaweed forms are, isn't it convenient you can photograph them instead of trying to trace and draw the whole thing? And so this interest in light writing is part of what inspired Talbot, as he was one of these early experimenters with photographs. And so he wrote about how cyanotypes are art that can help create this drawing without the artist's pencil itself. And so William Fox Talbot was friends with the father of Anna Atkins, and Anna Atkins was the daughter of this scientist who was part of the Royal Society in England. And at the time, women weren't really involved in this, or weren't encouraged to be involved in the sciences, but plants were okay. And as a result, so were ocean plants like seaweed. So you could go to the coast and collect them. And so she was really into collecting seaweeds, and through this connection with her father, was able to learn about cyanotype photographic processes at a really early time. And so it wasn't Talbot, it was actually her that created the first bound book of cyanotype photographs of seaweeds. And so as you can see today at the festival with all the beautiful cyanotype art, seaweeds have a really special place in the history of photography because they were in this first book. And so here are a couple of her specimens here. There's actually only 12, she made 12 copies of this book. A couple of them survived, and you can look at the New York Public Library if you'd like to see all the nice digitized versions of some of these. This is a photograph I took at Seabird to contrast what some of Anna Atkins' cyanotypes look like compared with the dried specimen. And so you can just see that with her images, the light passes through the body of the kelp in these nice ways. So if you have a more translucent kelp, then you're getting these fun shadow effects. Now, she also did something really cool. And so for me, as someone who studies media, I really like this part. She appears to have used seaweeds themselves to create the letters of the title of her book. So British Algae Volume 1, and then here you can see British Algae Cyanotype Impressions. And she seems to have used sort of a calligraphy. So you can use cyanotypes not only to create images of things, but also writing too. So she probably had some sort of glass plate that she used calligraphy and ink on in order to create the image on the left, and then on the right, we have this just gorgeous lettering. So it's like the algae are authoring their own book, right? Like they're participants in creating this book. So of course you can create cyanotypes yourself fairly easily. You can use either sort of pre-prepared paper, or you can buy the chemicals, potassium ferricyanide and ammonium, ferric ammonium citrate. Combine these together, it's good for a little while, and then you can dye different substances. So either your own paper or your own fabric. So I ran a workshop on environmental media with a friend once, and we had fun kind of stringing these cyanotypes up. And they're non-toxic, so they're great for kids too, and they're developed in water. So it's a really easy activity. So this also works really well with cloth. So here's my fun demo, which is that this is not very good, but this is a photograph too. So you can take like white cotton fabric, dye it with the chemicals, and then try to put the seaweeds on. I learned really quickly that this is actually hard because it starts exposing as soon as you lay it out on the sand, and then you got to work really quickly to put all the seaweed on top of it and in an arrangement you like, because it's exposing as it goes. Also, if you pick seaweed that is wet, it'll also start self-developing as it's lying on the image. So you can either be a little bit more precise and pick dried seaweed, or you could take a wet seaweed and just have fun with the effects of it, sort of staining little areas at a time. So I experimented, I made this quilt. I'm not very good, but if there's any quilters in here, this is a super fun project. And you can kind of sew them into different forms. I don't know if Josie Iselin is here at the festival today, but I've been really inspired by some of her art. She uses a flatbed scanner to scan the bodies of seaweeds and then makes these gorgeous, colorful prints. I think of this as halfway between a cyanotype and a digital photograph because it's sort of cameraless. Even though the scanner bed is scanning and it's a digital mechanism, the seaweed has to fit on the scanner. And so that's the same with cyanotypes as well. They're scale dependent. The seaweed has to fit, otherwise it's going to hang over. And so in that way, you can't sort of do the zoom effects that you would with a digital camera. So this is sort of a fun, I think, intermediary stage. So as I was looking into seaweed media further, I also found some crazy agar print photographs from a grad student at UW, Russell Marks. And so he took a selfie with algae. So if you think of seaweeds, they're photosynthetic, they're a type of algae. It's like their photo paper. So he treated this algae type as a kind of photo paper, and then took a sort of a photo negative in an enlarger, projected the light down onto the petri dish, and then was able to capture this image of himself. And then he froze it using some, I think he poured resin into it to fix the image. The image on the right, which I think is just some pine trees with maybe a wasp catcher. I'm not sure why he picked this subject, but this species of red seaweed is actually from the ocean. And so I thought, there's the ocean connection to algae further. It turns out seaweed is also good as a developer. I don't personally know a lot about darkroom photography, but people like Melanie King and others have been holding these photo developer workshops using seaweed as one of the ingredients to develop pictures. So think about this, you take a picture of seaweed and then you use seaweed to develop the picture. There's something just really fun about this. There's a long list of how-tos with this. It involves, I think, some type of vitamin C and maybe some salt crystals and other things, but I just show you that this is, these methods are available to Google as you please if you're curious. And then there's this film. So using seaweed developer methods, Julia Parks made a film called Bladder Rack, which is about a type of seaweed that she gathered. And you can see her on the left collecting some of the bladder rack and getting ready to put it in this boiling pot of water, along with some of the other ingredients I just mentioned. And so you're watching this film. This is available to watch for free online if you're curious. And you suddenly realize she's developed the film itself with seaweed. And she even hangs the film out to dry, like strands of seaweed on the line over on the right. And I just thought this was a beautifully gorgeous moment where the medium and the content were sort of self-reinforcing each other. And then finally, Sam Nightingale, who's an artist in the UK, started experimenting with seaweed photography, not because so much of the ingredients in the seaweed, but because it is coated with salt water. And so you can do a type of photography by placing seaweed directly on a certain type of photosensitive surface. And it's the chemistry, not the light, that actually creates the image in this context. And then finally, going back to more familiar images of kelp, perhaps, I think it's significant that in satellite photography, you can get images of seaweed because the seaweed makes itself available. So we have these wonderfully large bladders. This is a photo I took off Anacapa Island here in the kind of nearby, and the bladders hold it up. I've even seen cases diving where the seaweed has used its holdfast and latched onto some rocks, but they were too small. So as it grows bigger, you start to get this floating island effect where the rocks get pulled up and they're just kind of hanging there buoyant. I think that was really, it's like science fiction, right? You just see it hanging there. But it floats near the surface, and also chlorophyll A helps seaweed to be detected by infrared satellites. So here we have the seaweeds giving us a boost. We can image it better because it's participating. So as you can see with projects like Floating Forest, which I'm not part of, you can sort of highlight these patches of seaweed that are based on satellite images, and it's because the seaweed is floating there. So this works really well for giant kelp, but also for, in the Caribbean, sargassum, which is currently a problem. So that's it for today. I just wanted to tell you what I had discovered with all these fun interactions between seaweed as media and also the way it participates in its own photographs of itself. So I guess you could call these kelp selfies. Maybe that's what I'm going to say. I'm going to call them kelp selfies. All right, thank you.
Anybody have any questions for Melody? Yes.
Hi. So for the cyanotypes that Anna activated, at the time when they considered more an
would have been more of an art project. So that early stage in photography, like the techniques were widely varying all around. And I think this was even before daguerreotypes. And so they were not, this wouldn't have been like a widely used process. So for her, she was probably in her backyard with low, sort of lighter British sunlight. So she wrote it took like 5 minutes for her, and here it takes 1 minute to develop your photograph. So yeah, it would have been something a lot more niche. And so she was kind of only rediscovered recently. And so you can see lots of sort of fun pop articles now that really celebrate her work and just the beauty of this. She also thought of her book as a companion to existing seaweed guides at the time, which would have been based on either pressings or illustrations. And so hers, she thought of the cyanotypes as actually a very accurate method of getting the form on paper, and again, as like a companion to a different kind of field guide. So thank you. Yeah.
Question? I'm wondering if, if you will have this PowerPoint available online for those of us who didn't take notes and wish to, or if maybe this is an organized thing that you'll have videos online.
So we are recording all the talks, so I think we're gonna put them on our YouTube channel.
Okay,
So
so amazing work, so much fun. Also, Erika, phenomenal. Just so excited to be here with my fellow peeps, and beautiful work.
Oh, thank you. So I'll just mention briefly, I actually have some of this work published as a book chapter. And so if you email me, I'm happy to send it over to you.
Okay,
yeah.
Thank you.
very cool. Okay, last question. Anybody?
All right, thank you.
Okay, so next we're gonna hear from Dolinar, Reef Check, California, Citizen Science Reef Monitoring.
Well, thank you so much, Simona. I want to thank all of you for coming today. This has been so awesome for those who went last year and went virtually. It was a great introduction, and now see it become the real life event it is today is really awesome. So thank you for everyone for coming. My name is Dillon, and I am the Southern California Volunteer Coordinator for Reef Check. And before I sort of start talking about what Reef Check is, I have to dive in to talk about what citizen science is. And citizen science is going to be science that can be conducted by the general public. You don't necessarily need to be someone with a professional academic background to come out and collect data. And typically it's going to have 3 goals. The first one is, I can't really emphasize this enough, we want it to be open to lots of people. Typically with citizen science, the more diverse array of folks who come out and help us, the better data we can collect. Also, I want to work over large areas of space and do stuff over long time periods. You want to be able to get that long-term monitoring data to come back year in year and see how are things changing if they are. And then lastly, we don't want our data just to be sitting, in a computer or on a Google Drive. We want it to be out there giving information to management agencies and allowing them to make scientifically informed decisions. And citizen science puts us in a pretty unique position because we were able to conduct our research, but it really puts us at the forefront of community engagement. Oftentimes the stakeholders who are out living and working in the areas, they come out and work with us, and they want to be able to be like, what's going on in, literally my backyard. And because of this, we have a very unique opportunity to work with education, both with younger folks, like we can see here, as well as older people who come out and volunteer with us. We have volunteers all the way from, I think, like 18 to people well in their 70s that still come out with us. So lots and lots of people. So let's talk about what Reef Check Foundation is. It was established in 1996, pretty much with coral in mind. That's what they were going with. And then, years went by and they're like, hey, kelp forests is pretty cool too. Let's look at those. And in 2006, Reef Check California was established. And basically what we do is we provide data and information for marine management agencies to sort of make those scientifically informed decisions. And we do this by monitoring rocky reefs and kelp forests using standardized protocols that we basically keep the same every single year. This is a picture of me diving really close to here at Refugio State Beach. I hear I'm asking a sea urchin where it thinks all the kelp went, and it didn't know, so I kept swimming away. We look at 4 main things when we're surveying. We're going to survey 35 different types of fish, 34 species of inverts, 5 species of native algae, and now 5 species of invasive algae. Then also, we're going to basically look at what's going on with the seafloor. Is it flat? Is it steep? Is it rocky? Is it sandy? What's going on with that? So where do we do all this? Here is a map of California, and we go all over the place. We've been doing this for 15 years, and we have 149 sites now, all the way from San Diego and Point Loma to basically the Oregon border. And now we're extremely excited. In the last few months, we've expanded into Oregon and Washington. So and then hopefully, who knows where it'll go beyond there, because we actually have a Baja coming up next year. So really exciting times. So let's tell a few seaweed stories. That's kind of why we're here. The first story I'm going to tell is about an invasive species that we were not able to contain. And then the other 2 are about species that are invasive species that we did contain, but did it differently for each of them. So the setting of this first story is a place that I love. I'm sure many of you do too. It's Catalina Island. And you can see here that all of these little orange pins are sites that Reef Check dives and surveys every single year. And here in the blue and the red and the purple as well, those are going to be marine protected areas. And marine protected areas are areas that they might have limits to fishing or dumping. And they basically just, it's not like people can just go out and fish whatever they want to. There are some level of restrictions. And so the 2 main characters in this first story are going to be giant kelp. It's going to be our native species that we have here. We heard plenty about it today. It's awesome. And it's also a foundation species, which means it can basically form, it's important for habitat forming for many of the other species that call our rocky reefs home. And perhaps if you will, our antagonist of this story is going to be an invasive species right here, Sargassum horneri. And what it can do is displace some of the native species, particularly giant kelp, when times of like disturbance come. So say a big storm comes in and hits Catalina Island. What can happen is that might rip up a lot of the kelp, and the sargassum might be a little bit better at like taking over that now available real estate than the giant kelp might be. So we wanted to know where's the giant kelp doing better, and can these marine protected areas kind of harbor more areas of dense kelp? And when we look at this, it's honestly not really what we thought we were going to see. We look at it, and here we have kelp density on the side and year on the bottom. And so what we're seeing is that the red line are the marine protected areas, and the blue are the non-marine protected areas. And there's really not a big difference, and this puzzled us until we thought about some of the nature of Catalina Island. And when we look at it, we have 2 main parts. We're going to have the front side and the back side. The front side is going to be the side that's facing LA, like Hollywood over there. So it's going to be much easier for, fishing vessels and maybe like just people to come to the front side and maybe just even do a day trip and go back to, LA, San Pedro afterwards. The backside is a lot harder to get to, honestly. It's subject to the open ocean. There's no protection for it. So it can get hit with larger swell. It's also a little bit colder back there. And so we wanted to look at what happens to the giant kelp when you're just looking at it on the front versus the back. And so here, just looking at the front side, we're looking at how the MPAs might affect it. And what we see is that there's barely any giant kelp outside of marine protected areas on the front side where more people spend their time. But we do see that there is kelp inside the MPAs still. When you compare this to the backside, it's very interesting because we see there's not really, this is what was driving that first graph. We don't see a difference inside and outside of the marine protected areas. So this is really interesting because what it can tell us is maybe there's less anthropogenic disturbance going on in the backside because it's harder for people to get there. And so you can just see this right here. these are just the trends that I was basically talking about, where outside of the marine protected areas in the front, barely any kelp, a little bit of a difference, and then the backside, they're basically happening in the same ways. So this prompted us to ask, like, is the kelp denser on the front side or the backside? And what we see here is that pretty dramatically up until 2019, that there was far more kelp in the green, which is the backside of the island, or far denser kelp, I should say, at our sites. So we heard about our hero of the story. What about maybe our villain of the story, our sargassum? And what we can see is that across the whole island, there's typically a lot more sargassum inside the blue, which is the non-MPA, so areas that get no protection. The invasive species does better there than the areas here in the red that are inside the marine protected areas. So we asked the exact same question that we did with the giant kelp. Is there more of this invasive on the front side or the back side? And so what we see here is that it's the opposite. We see that there's a lot more of the sargassum on the front side, and there's barely any on the back side. So this is the opposite of our giant kelp, which does better on the back side, and the sargassum does better on the front side. So let's hear about this other story. This first one takes place in Newport Harbor. Many of us might know what this is. It's something that we don't want to see. This is an invasive species called Caulerpa prolifera, and it was seen here in Newport Harbor, and it was discovered by some divers. And they came across it, and they looked at it, and like, this does not look like anything I'm used to seeing. And they went around, and they tried to look for it a little bit, and time went by, they're trying to ID it. And by the time that it was successfully ID'd, it had basically become this massive carpet and was difficult to remove. Some of the people here today actually helped in those efforts. So it was like it's doing well now, but it required a lot more time and resources because it took more time for it to be ID'd. Let's compare that with this. This is a juvenile sargassum horneri that was clear on my computer up here. But basically, if we look at this, for many of us in Southern California, we see this almost every dive, and we don't think much of it. This picture was actually taken at the breakwater in Monterey, about 270 kilometers north of its previously known invaded range. And luckily, the diver who saw this was a reef check diver. And they have basically undergone the training to be able to know what they were looking for. And when they came across this, they're like, I know this isn't supposed to be here. And they went and told the proper management agencies, and this was successfully removed much easier than the Caulerpa was. And so you might be asking now, like, how can I get involved with this? Here we have, you can see, we like to have a lot of fun when we're out there. So basically the way it works is our classes, you learn lots of great stuff. We talk about marine ecology, oceanography, and fisheries, MPAs, all this really fun, nerdy stuff that we really, really dig. So it's usually done over a 4-day training. We have 2 days, I'll get out of the way. We have 2 days in like a classroom or pool to sort of practice our skills. And then 2 days in the open ocean, perfecting those and really putting them to use. And we, every single year, we do annual recertifications. We go out in the spring and we practice to make sure that our skills are ready to be put to test every summer and fall. So with that being said, I want to thank the volunteers who make this happen. You know, I didn't individually collect all the data out there. In fact, I barely collected any of it. It's all the volunteers who come from all kinds of walks of life, from all over the state, who come out with us on their weekend and their time to come provide this data that we use and the state uses for many reasons. And so I actually want to thank 2 of the volunteers. They don't know, they're 2 great people in the back, Jade and Ivan. They've been at Reef Check longer than me and they've contributed a ton. So I want to thank them personally. And lastly, here's my information. If you have any questions, if you're interested in joining Reef Check, learning more about what we do, I'm happy to talk.
For Dillon.
Yes.
Go ahead.
What are your feelings about using the offshore rig this week?
Oh, that's a great question. So I think that there's definitely promise, like if you're talking about like if there's a decommissioned rig that, yeah, I think that those can provide like they have promise to basically be like an oasis for different things. I know my friend was just diving some of the rigs in the Catalina channel and saw like Garibaldian animals that we wouldn't expect to see way out offshore. So, you know, if habitat loss is a problem, I think that basically using some sort of human-made structure that is no longer in use to supplement that loss would be a benefit.
Yep.
Hi, I was just wondering if you ever would authorize the divers to remove anything, like if they saw the sargassum, are they able to remove it, or you just leave it alone or sell it?
Yeah, so for us, that's a big no, unfortunately.
Okay.
We take the pictures, if you can, get like extremely precise GPS points, and then give all the data to the management agencies, and they're going to be the ones that basically call the shots from there.
Any last questions? Okay, well, thank you, Dillon. Microbes and seaweed. So we're gonna hear from Laís. Okay, take it away.
Thank you so much. Hi, everyone. How are you doing? Great. My name is Laís. I'm a PhD candidate at San Diego State, and I'm transitioning from a microbiology lab, the Dinsdale lab, to Dr. Matt Edwards' lab, which is a kelp ecology lab. And so I'm fairly new in the seaweed world. So being here in this festival, for me today, it's really important because I am learning so much from everybody. And let me tell you a story about microbes and seaweed. So who runs the world? I wish it was girls. I wish it was seaweed, but it's not. It's actually microbes, because they're the ones that are providing essential services to all life forms. They've been around on Earth much longer than anybody here. They're really good at transforming elements, so making things that weren't available before to one life form to the other. Particularly, very importantly, nitrogen. They're very good at bringing the element from the atmosphere into the biological systems into several different steps. And they break down organic matter, so they're decomposing things, and they're involved in the production and consumption of oxygen and carbon dioxide, among many, many other things. This is just to name a few. We've always been looking to microbes recently too, to help us to clean up our mess. So there's a lot of research being done on microbes to help to degrade plastics, potentially oil-derived products and things like that. And so in general, microbes are really here for us. And I did a really quick Google News research. I just typed microbiome yesterday, and there's plenty of articles, some of them saying that the microbiome can be used to monitor pregnancy risk, that the human microbiome is, that bacteria can help you to grow your muscles. And essentially that microbes are the secret of a long and healthy life. Not a big deal, right? Like, no pressure. And the cool thing is that microbes, they provide similar services to different hosts. So whether you're looking at a human and their microhabitats and their microbiome, or if you're looking at a plant, essentially the three major ways that microbes help their hosts is one, making their reproduction healthy. There's a study that showed that couples that went through in vitro fertilization, the ones that were successful had specific bacteria that potentially is correlated to their reproductive success that couples that did not get pregnant have. So microbes can even be related to that. Microbes are very famously related to nutrition and metabolism in our gut. I think everybody have heard about the gut-brain axis and how microbes can modulate how much nutrients you're getting, our mood, and even to recover from trauma, they're related to that too, apparently.
And microbes can also help us to be protected from stressors, either biotic stresses like pests and pathogens, or abiotic stresses, especially in the case of plants. They're very effective to help them to survive heat stress and drought, the microbes in their roots. And what about seaweed? So in the seaweed microbiome, they're performing also similar functions, and that's what I'm gonna tell you about today. They do get involved in reproduction, nutrition, and protection from stress. And for nutrition and protection, instead of being compartmentalized like plants, where we have the leaves and the roots, in seaweeds, that's gonna all be happening on the blade surface. So because the whole mass of seaweed is not actual real root, in the sense that's not the part of the seaweed that's absorbing nutrients, that's happening on the blade. A lot of really interesting things for a seaweed is going to be happening on the surface of seaweed. So they host a very complex, diverse, and thriving microecosystem on their blade, where there's a resident portion of the microbes, a more stable portion of them, that it's going to be operating as a biofilm. But there's also microbes that are more transient. There's also going to be more affected by what are the microbes in the water column, what the conditions in the water column are. And microbes are going to be secreting compounds, and that's going to be affecting the host, the seaweed. And seaweed also secretes things that affect the microbiome. So it's a very complex and very well tied and integrated micro ecosystem. So first topic, reproduction. Healthy microbiome means a healthy seaweed babies. So there are several studies showing that specific bacteria help seaweed to settle. So to go from the free living stage into the substrate where they're going to develop. They're also essential for their morphogenesis. Those pictures show, the picture on the left shows, I believe it's ova that has been, the bacteria has been removed. And so their morphogenesis was compromised. And as soon as they added that bacteria back, they were able to recover and develop properly. So that leads us to questions such as, could we use microbes to increase seaweed recruitment and survivorship? Also, there's evidence that the microbes in the water column of urbanized areas can be detrimental to kelp babies. So the study was actually done by my lab colleagues at San Diego State. And they show that water collected from Point Loma near San Diego, which is a highly urbanized area, when they increase the concentration of the microbes that naturally occur in the water column, you get very stunted, unhealthy gametophytes of giant kelp. And at low concentrations, they do really well. So we could be affecting kelp recruitment just by altering the microbes that are in the water column in our coastal systems. In the topic of nutrition, microbes provide essential nutrients to seaweed, such as nitrogen that we talked about. Some seaweed have nitrogen-fixing bacteria, and it's actually pointed out as one of the keys of success for some invasive seeds, such as caulerpa that Dillon talked about. So apparently caulerpa has nitrogen-fixing bacteria, which helps them to thrive in oligotrophic waters and have competitive advantage. But if you isolate those nitrogen-fixing bacteria and you apply to Gracilaria here, for example, it boosts their growth. So it improves their growth, even though they're not their original microbiome. You can manipulate the microbiome to get yields that you didn't have before. Again, leads us to another interesting question. Could we use microbes to help seaweed to grow faster or better? And there's also some research showing that for kelp to cope with heat stress. If you increase the nitrogen in the water column, even though they're still being exposed to heat stress, there is a buffering effect of that stress. And so another key question is, could we use microbes to help kelp to get more nitrogen so they can cope with a warming ocean? And then finally about protection from stress and pathogens and parasites. Opportunists just like encrusting bryozoans. I think kelp farmers know them very well. And how much do you love them, the bryozoans? Nobody loves them. Because these guys, you can see here, they cover the seed tissue. So if you're trying to photosynthesize, that can be very challenging. And for growers, you can have a very high percentage of their biomass that it's not the seaweed, it's other things growing on it. So there's some studies, there's a research paper actually here from a group from UCSB that points out that bryozoans may be altering the seaweed microbiome by introducing their own microbes, and that could be a mechanism that's causing dysbiosis and helping them to encrust even more on the kelp tissue. So could we manipulate the seaweed microbiome to avoid Fowler's disease and parasites? That's another very interesting question. So when it comes to seaweed farming, so applying all those questions that I was telling you about to the setting, we really want to know where do microbes fit here in the system, because it's considered the next frontier in seaweed aquaculture. I wasn't the one that said that. There's a whole review paper pointing out and saying that the microbiome should be seen as an extended phenotype of macroalgae. So that leads to our project that's called Micro to Macro, that we're here to leverage kelp aquaculture from micro to macro scale using the microbiome of macroalgae. So got it? Micro to macro. This project is super brand new. We recently just got funded, approved in the beginning of the month. So we haven't collected anything, we've just been continuing. Thank you. It's a really cool NSF grant that's called Convergence Accelerator. And it's not the typical research grant. This one is very applied to business. They have specific tracks. And the track that we got awarded in is called Networked Blue Economy Research. So there's this big investment to push for human design science, where we want to use what we know about the oceans and specifically about seaweed and seaweed farming to help our society in more sustainable economic activities. So the first goal of our project is to form a network of seaweed farming, because this grant is so centered to help growers. So essentially, we want to do science to help people, and specifically in this case, kelp growers or seaweed growers. The second goal is that we're going to characterize the seaweed microbiomes of a lot of different species across different locations, compare from farm seaweed and natural seaweed beds. We're also going to do experiments to test for microbiome manipulation. We want to identify and address which are the microbial players that can lead to targeted seaweed traits, depending on which seaweed industry and what you're interested in, if it's biofuel, if it's food, if it's livestock, fertilizer, you name it. So different seaweed farms are going to have different targets, and maybe different microbes can help growers to achieve those targets. We're going to test different farm designs that could support a beneficial seaweed microbiome. And we also have a very strong social and political part of the grant project, that is to streamline the permitting process that I'm sure if you've worked with it, you know it's not fun, it's very hard. And we want to integrate stakeholders and help to develop this industry, especially on the West Coast, and especially by involving indigenous groups from Alaska to San Diego. Oh, and right now we're on phase 1 that lasts for a year, actually 9 months. And then hopefully, if we pass the Shark Tank thing they have at the end, we hopefully get grant for 2 more years in that space too. And that's when things can actually happen better. The core team is Dr. Matt Edwards from San Diego State, he's the PI, and then Tori Polizzi from Sunken Seaweed, he's around, he did the cooking demo, he's an amazing collaborator. He's the co-PI, myself and Leslie, also from Sunken Seaweed. We are the core team. I am just so lucky to work with these people. And we have, for now, these collaborators, and we're working across some different sites in Alaska, some sites in California. We are looking for partners because that's what the grant's for. It's supposed to be very integrative and collaborative. So we want to talk to everybody. We want to bring more people and talk to us whenever you want. We're available, we're around. If you know Leslie, you know Tori, they're around, go talk to them. So yeah, hopefully just the take-home message. I hope you go home knowing that the seaweed microbiome can support seaweed babies. It can bring food to the seaweed table, and it can protect seaweed against opportunist and stress. And all of that is still an untapped potential to boost seaweed farming. So there's a lot of unknowns about how we can apply these concepts. And that's what we're very excited to start doing research on. And thank you so much. If you would like to collaborate, this is my email, lima@scsu.edu. And yeah, come talk to us. Thank you so much.
So we'll go ahead and have some questions right here.
Really excited about your work. We're big fans of Landon Ingham and the Soil Food Web. And so there's a growing appreciation of a differentiation between probiotic and prebiotic. That you know, we eat yogurt and sauerkraut, you know, to enhance our gut flora, but a lot of those bugs don't actually survive in our gut. So they're kind of like food for the microbes in our gut. And so we're getting more towards looking at what are the appropriate foods so it enhance the appropriate communities that you know, in agricultural situations, the microbes are there, they're just eat the right food. So I was kind of wondering about, you know, probiotic versus prebiotic in the environment.
You mean for the human
purpose
or for seaweed?
In the seaweed.
Yeah, so there's, I don't think that I know of that I've seen any research addressing specifically that. But for our grant, for our proposal, we want to work more towards prebiotics because we want to make it very applicable to farmers. We don't want to necessarily have an isolate and then a concoction of microbes. We are going to test that too. But if we can find ways that farmers can manipulate certain conditions and get to an ideal microbiome, that's better, that's easier, that's more streamlined than having scientists doing a lot of isolates and streaking plates and things like that. So yeah, that is super interesting. I was talking to Doug, I don't know if he's around. We were talking to him yesterday, and he was throwing some ideas about certain mixes of seaweed to the abalone and how much that affects their growth and their health. So we were, for example, wondering of sampling the microbiome of different compositions of feeding and then seeing which compositions are the best before even thinking about creating probiotics to feed them. So it would be more in the prebiotic kind of kimchi, kombucha, not even kombucha, but prebiotics idea. Yeah.
Thank you. Are there any other questions? Okay, really fascinating. Okay, next up we have Emily Bews, and she's going to tell us about farming sea lettuce for bioremediation and sustainable food production in San Diego. Take it away.
Can you hear me okay? Yeah. So I just want to say really quick, a big thank you to all the people who put together California Seaweed Festival and everyone who's participating. I've learned so much today. I'm having such a great time. But yeah, I'm here today to talk to you guys about farming sea lettuce and seaweed aquaculture, specifically for applications in bioremediation and sustainable food systems. So to start off, we have a little bit of a background in this at this point, but basically seaweed aquaculture is a globally widespread practice. However, it is relatively young in the United States. And traditionally, industries that we rely on for products and goods that are essential to our way of life, such as products that are a result of commercial agriculture, for instance, often require significant land use and nutrient inputs, as well as large volumes of fresh water to facilitate production. However, seaweed aquaculture, on the other hand, can provide us with more sustainable alternatives for these goods without the strain on limited land and resources. So as you've seen here today, we can produce really amazing food with seaweed, animal feed for livestock, fertilizers that can replace the need for synthetic fertilizers, as well as bioplastics and biofuel that can help us transition from fossil fuel-based production of goods. Seaweed aquaculture can also provide many environmental benefits, some of which are pollutant and nutrient uptake. So urban runoff is a huge problem that affects watersheds and bays and estuaries that are situated in urban settings. As water travels across urban environments, it travels across impervious surfaces like concrete and asphalt. And this is where it can pick up a ton of different pollutants and contaminants. So this can include fertilizers and excess nutrients, pesticides, toxins, bacteria, waste, oil, grease, the list goes on. One of the main effects can be seen through harmful algal blooms. That's an example of how bays and estuaries, specifically bodies of water with less water exchange, feel the most severe impacts of nutrient pollution or eutrophication. So harmful algal bloom occurs when algae or cyanobacteria is able to grow unchecked because of an excess of nutrients in the system. And as this cyanobacteria or algae begins to decay, this can eat up all the available oxygen that's in the water. At a certain point, there's not enough oxygen below the surface to support life. And on a large scale, this can be called a dead zone. There's also issues with harmful algal blooms being severely toxic to human and animal health. So we have a dire need to fix the conditions that are affecting the ecosystems that we rely on. And seaweed aquaculture can provide a means by which we can try and fix these conditions. So bioremediation is the use of living organisms in order to help uptake contaminants such as dissolved hydrocarbons or metals, as well as nutrients such as nitrogen and phosphorus in order to improve living conditions in a given environment. And knowing what we know about seaweed aquaculture having the potential to uptake contaminants, we can specifically use the culture of seaweed to improve water quality and living conditions in an environment. So now moving on to sea lettuce, what is known as Ulva lactuca. This is a globally distributed green macroalgae, and it's been widely studied for its uptake potential in different situations. And while there's been a lot of research done on its uptake potential in general, there's been little research done on the use of Ulva lactuca specifically for bioremediation under conditions of low salinity and elevated nutrients. So that's something you might expect after a major runoff event washes pollutants into a bay or estuary, also freshening the surface waters. So we decided to study this as a perfect candidate for potential bioremediation uses. So San Diego Bay lies in a highly urbanized setting, and it's periodically affected by anthropogenic pollution, boat traffic, and recreational traffic.
This is one of the most polluted bodies of water in the United States, which is pretty unfortunate. But in an effort to attempt to address the water quality issues that we face in San Diego, sunken seaweed, which you've probably seen around here today, Leslie Boomer and Tori Polizzi, in conjunction with the Edward Lab at SDSU, launched a pilot scale bioremediation farm in the Bay. And this research was supported by the Department of Energy, whose interests lie in capturing those excess nutrients and researching alternative energy sources. So we set out to try to determine how a major runoff event could affect conditions that would impact sea lettuce's health and productivity. So in order to address this, we posed the question, would farming ulva be a viable option for bioremediation in polluted coastal watersheds, specifically when you're expecting drops of salinity with the input of elevated nutrients? So this research was conducted at the Coastal and Marine Institute Laboratory in San Diego, California. In spring of 2020, we set up 18 tanks stocked with ulva, and I'll go a little bit more into the treatments we added later. But basically, this helped us gain a more comprehensive understanding of the conditions that might affect ulva's growth and productivity, specifically when it is used for bioremediation. So here's a little mock-up of our design. I filled some jars with some pictures. But essentially, we set up these tanks in spring of 2020 in a temperature-controlled room and spiked each treatment with different levels of nutrients. But first, we decided on a high and low salinity level. So the high is representative of what you might see in San Diego Bay just on a typical day, while our low salinity represents what it might be at certain areas in the bay, like at the lowest salinity it would be during a major runoff event. And then the nutrients we decided upon, for our low nutrients, we did nothing, it was just ambient seawater. And for our mid and high level nutrients, we created a solution out of a commercial fertilizer that is very rich in nitrogen, phosphorus, and potassium. So this middle level of nutrients was similar to what you might see in the bay when pollution is washed in through runoff. And we chose a high level just to see if there was a limit to ulva's tolerance under different salinities. Okay, so a couple graphs to show you guys, bear with me. We measured growth about once a week during the course of this experiment. And while ulva did well in all of our treatments, it survived, it grew. Growth was slightly lowered under low nutrient conditions, and it was at its optimal growth in medium nutrient conditions. So this suggests that those mid-level of nutrients, which I mentioned before is representative of nutrient pollution in the bay following a runoff event, would be most beneficial to ulva's just general growth. And while this was noticeable difference between different nutrient treatments, it was consistent among salinities. And this indicates that salinity was less of a driver of growth than nutrient presence was. So here we have quantum yield. This is sort of an abstract measure because it's something you can't necessarily see right off the bat. But quantum yield basically tells us what's going on with ulva's photosynthetic efficiency. And a value above 0.6 indicates that ulva's photosystems were not under severe stress and were functioning well. So as you can see here, all of our values lie at or above 0.6. And this indicates that ulva, regardless of the treatments we exposed them to, exhibited stable photosynthetic efficiency. All right, and then we also, at the conclusion of the study, decided to determine net oxygen production sort of as a proxy for photosynthetic performance. So while it was slightly lowered under low salinity, you can see that the addition of nutrients to the seawater did elevate oxygen production. And we found that similarly to growth, oxygen production was at its greatest in our mid-level of nutrient treatments versus being lowest in our low level of nutrient treatments. And finally, we did some tissue analysis for nitrogen and phosphorus. We found that nutrients were a stronger driver of nutrient uptake in general. The presence of nutrients helped drive this phenomenon of nutrient uptake and sequestration. And the addition of nutrients to the water elevated this uptake. Oh, and that I just repeated myself, but nutrients were a stronger driver of uptake than salinity was in this case. So our key findings, we found that while low salinity did have a negative effect on the growth of our ulva, the addition of nutrients really eased this effect. And though salinity had less of an impact than nutrients did, it was still a factor in this. So future directions for bioremediation research. Our findings will help aid resource managers and policymakers as well as farmers meet water quality improvement goals and try and determine new locations that this would be effective for use of bioremediation at. This can also help farmers reduce costs associated with operating their farms. So knowing that ulva, in this case, would do fine in lower salinities, farmers don't have to worry about lowering their entire farm structure in case a drop in salinity might impact their productivity. More research should be done on the biotic and abiotic factors that could influence bioremediation operations in the future. Okay, so now transitioning to the second application of aquaculture I introduced at the beginning. Integrated multi-trophic aquaculture, or IMTA, which I'll call it from here on out, is the practice of utilizing organisms from different trophic levels. So if you're not entirely familiar with that, you can think of a food web and how nutrients and energy travel through that food web, starting with a primary producer and going up to consumers and predators along the way. So using as much of the resources available as possible, so say you have a fish farming operation, there's available nutrients that aren't being recycled when you're just raising fish. But if you're introducing seaweeds and invertebrates, you're able to take advantage of other things that are available from that system. So where I work currently at Hub SeaWorld Research Institute, we've had 2 projects so far investigating the applications of IMTA. Our first project occurred in Carlsbad, California, and our second project is happening right now in Mission Bay, San Diego. So here you can see what was going on in our Carlsbad operation. This is some of the white sea bass that we use to feed our macroalgae nutrients. And this is a flow-through system, meaning that water went from the white sea bass tanks, transporting dissolved nutrients and CO2 down to our macroalgae tanks. And once the water hit the macroalgae was able to take advantage of those available nutrients and CO2, and the water was returned back to the lagoon we originally drew from in the first place. And here is a closer look at some of the various seaweed species that we used. If you were out there earlier today, you were able to possibly try some of the gracilaria that they had for the cooking demonstration. Gracilaria sort of has like a light, crispy taste to it, sort of salty, like as you would expect for a seaweed. And same with ulva. Ulva is a bit of a lettucey flavor, still crisp and salty when you bite into it. And then dulse, which you've also heard about talk today, sort of has almost an arugula flavor, I would say. And it's pretty well known for tasting like bacon when you cook or fry it. All right, and so transitioning to the more complex system we have in Mission Bay currently. We're not only utilizing fish and seaweed, but we're also utilizing invertebrates for this project. Basically, fed fish are the main source of nutrients in this system, as the only external input is feed for fish. So as the fish excrete waste, this comes in the form of dissolved nutrients and solids. So our sea lettuce, as I mentioned before, is able to eat up all those dissolved nutrients and also introduce oxygen back into the system so the fish have a constant source of oxygen. And then the mussels and sea cucumbers are able to take care of the solids in the system. So the mussels filter out those suspended organic particles, where our sea cucumbers are able to eat all the solid fish waste that is left behind. And so one of the main differences from this second phase of research compared to what we were doing down in Carlsbad is not only the introduction of invertebrates who can take care of solid waste, but also this is all happening through a recirculating aquaculture system. So this means that water is circulating continuously throughout the system, And this normally would require extensive filtration and as well as oxygen inputs in order for fish to be able to survive. However, utilizing IMTA sort of gives us a nature-based approach of addressing these requirements. So our macroalgae, as I mentioned before, is able to uptake those nutrients as well as re-input oxygen back into the system and assimilate carbon dioxide. Whereas our mussels and cucumbers act as sort of a solid waste filter and eat up all the nasty stuff in the water. So as we move forward and IMT becomes a more mainstream practice, we need to make sure that we're utilizing native species for this kind of aquaculture, especially when it's deployed in open water. This is to ensure the health and safety of neighboring ecosystems. More research should be done on sustainable fish feeds. So there's a lot of problems with fish farming right now. And one of the things is resource availability. When you think about it in terms of what they're looking for fish feed, like you need a protein and you need carbohydrates, Ulva has potentially shown a promise to be a really good protein replacement in fish meal. Some more research is being done on that, but that's some pretty exciting stuff, trying to make sustainable aquaculture even more feasible and sustainable. And then finally, as more research is done, biofiltration and system design will continue to improve and get better. And this can make integrated aquaculture a more feasible and very strong and important part of our global food systems. So I just want to thank all of these people up here. I want to thank the Seaweed Festival for having me. There's a few more talks after, so I'm excited to hear from all of those people as well. And let me know if you have any questions. You can find me afterwards too. Thank you guys.
For Emily. Yes, right here.
Yeah, I just have two questions. So, you mentioned that you use bypass, and you also use the yellowtail, and also
Yeah.
in part of your IMT system. So is that also not for output, but the gracilaria and the some area, or are you also switching from the fish species depending on the seaweed that we're growing?
Yeah, so we specifically have white sea bass up in Carlsbad as part of their hatchery operations. And we did different research trials trying to experiment with different flows and shading. And that's when we decided to employ some of the red algae, just because we were able to really control those conditions and see what was affecting it and what kind of product output we would have at the end of the day.
Any other questions back there?
Your initial research with nitrates, right?
Yes.
And how well did it translate over to the actual living system?
Sorry, can you repeat that?
Yeah, how did, your initial research done kind of isolation. How did it transfer into the living system of the ponds?
It's given us sort of, because it's a land-based recirculating system, so we have several different tanks set up. It's pretty complex. So that initial project that we did, just looking at fish and algae, sort of that two-tiered system, gave us some insight into how to improve the biofiltration efficiency of different kinds of seaweed. So just ensuring that they are actually uptaking enough and that it is sufficient biofiltration in order for the fish to, be safe and be in livable conditions. So it's sort of translated over in that way.
Yeah, so it's your first part of research to feed into the fish.
Yeah, we started off like a little bit of a smaller scale, just in terms of complexity of the system, using just the 2 trophic levels, just to get us started. And now it's kind of becoming more of a pilot scale, what you would want to see out in the ocean.
Sort of reminding me of John Todd's work, the living system.
Oh, yeah.
And he's a quaint. So you're working good. Thank you.
Thanks.
All right, well, thank you so much.
We have Julia Marsh. She has been designing all of our California Seaweed Festival logos. So fantastic. Thank you so much, Julia, for all your work with that. And so she's going to tell us more about seaweed bioplastics and regenerative design.
Thank
Thanks, Simona and Emily. That was so nice. Lovely talk. Hi, guys. This is going to be a little bit different than I think a lot of the other programming, a little less academic. But I would love to know in the audience, who's already familiar with seaweed bioplastics? Who hates them? Who's like completely opposed? Let's not do it. I won't judge you. But you will be invited to sit here and like the whole lecture will be just for you. Okay. Well, I'm going to talk a little bit about my background as a designer, how I came to be a part of this beautiful seaweed community, what's happening in the world of regenerative design, and then I'll talk a little bit about Sway. It's not a sales pitch. We only work with companies, so I'm not selling you anything, but I hope to sell you on some of these ideas. So I'm Julia. I am Julia Marsh, the second one. You can remember it like a swamp. I am the CEO and co-founder of Sway. I have a background in brand and packaging design, and I'm all about seaweed innovation. I also love the ocean, even when it has sharks in it. So that's me, and being very happy with the sharks. What I don't love is this kind of imagery, which I'm sure you're all very familiar with. What you see here is a whole lot of thin film plastic circulated in a bay outside of Indonesia. And the reason that these are especially devilish is that they float out of landfill into oceans. They gum up recycling machines. A lot of them can't be recycled: bags, pouches, mailers. They may even have a recycling logo on them, but they are lying. They're not recyclable usually, or not accepted. And they end up in our natural spaces for eons. And the reason this is so close to my heart is because in my capacity as a designer, I was the one responsible for bringing those thin film plastics to a brand. So no matter how beautiful the vision or the company or the team, I'm the person saying, all right, well, we got to package it. what are we going to put it in? And so I really came to think about what is the designer's role in protecting the planet and its people. And that's what inspired Sway, really looking at the interconnectivity of American consumption, this rising demand for convenience, and a whole lot of social inequities, especially that have been felt in these past couple of years with COVID. How can we design solutions for the plastic problem that are intersectional? So plastic problem is not isolated. It's highly tied to the climate crisis. We've got petroleum. The climate crisis feeds social inequity. Social inequity feeds the plastic problem. So let's design solutions that address all three of these categories. So I kind of went over the overview already, but I'm gonna have a little introduction, a little context. I'm gonna talk about some other materials that really inspire me. And then I'll do a bit about Sway and our potential impact. So just up front, we make bioplastics from seaweed. That's one of them. Those are my hands. Regenerative materials are really cool. There are so many. I'm just going to talk about a couple, just a few. I'm going to try to cap myself for time. We all are probably familiar with mycelium. This is a replacement for styrofoam, mostly known from the company Ecovative, which has pioneered this. Really cool because it's materials that grow themselves and then turn back into healthy soil at the end of their life. Companies like IKEA are adopting this. It's quite scalable. Another alternative that's relatively local, this is a company called Cruise Foam. They're based in Santa Cruz. They use chitin from shrimp waste and lobster waste. Those shells can turn into a replacement for styrofoam as well. We've got our folks over at Bloom that are using this algae that's been collected from fresh water. They're cleaning water, they're turning it into a usable substrate that's been used in Jason Momoa's shoes and Adidas shoes, and they're all over the place now. Those are my hands modeling again. This was a visit we made down to Guadalajara in Mexico to visit the folks at Deserto who are working with Nepal cactus to create replacements for leather. What we really like about Nepal as a feedstock is that it is infinitely scalable. It doesn't need a whole lot of fresh water, just relies on rainwater, sequesters a ton of carbon. It's very resilient to freezing temperatures. It grows 24 hours a day. If I wasn't a seaweed person, man, that's pretty cool. And then we've also got waste materials. So really love the work of Coffee Form that's utilizing the waste, the coffee grinds, turn them into reusable cups, kind of completing that coffee cycle. If you guys know the folks at Make Grow Lab, this is Scoby packaging. Who loves kombucha? We're in Santa Barbara. Yeah. I feel like we need to get seaweed kombucha at the next festival. All right, we're on it. So this is the top of the, you're familiar with the Scoby. They're making this kind of resilient film. It is water resistant. It's a microbial barrier. And it makes a great alternative to some of those plastic coated papers that you see. This is another one. Our friends over at Outlander Materials are making replacements for thin films from beer brewery waste. So the founder over there always likes to say, I get to hang out at breweries all the time and then make packaging that degrades into zero plastic. And of course, there's seaweed. So many of you may already be familiar with some of the more prominent seaweed bioplastic companies. Natpla here is based in the UK. They are primarily focused at the moment on these sort of condiment pouches and little beverage pouches called the Uhu, as well as some coatings for papers. And I know they have all sorts of different exciting products coming out. And then Lollyware, our friends at Lollyware, I see the Instagram live, so Chelsea, if you're watching, love you. This is, they're primarily focused on the straw, but I know they'll also be expanding into all sorts of other exciting seaweed-based replacements for thin or single-use plastics. And then there's us. So we're part of this ecosystem, this movement. It is not us alone on this journey. It's a collaborative opportunity to replace petroleum through all these different solutions at scale. And the reason that we love seaweed so much has already been discussed in detail today, but I'm going to get a little bit into it now. But what we do love about seaweed is its abundance. It's a generous material, and to me, it is the epitome of a regenerative feedstock. So I'm going to talk a little bit about our solution. In order to beat plastic, you have to do a lot. You have to be non-toxic and compostable. So if we want to succeed, we need a material that degrades rapidly in natural conditions, assuming that people are not going to flawlessly adopt compost behavior, right? So this material, if it's disposed in a natural environment, needs to degrade within 4 to 6 weeks. If it goes to a landfill, it needs to degrade in the same time span as maybe a fruit peel. If it goes to the ocean, it needs to not poison sea life. So that's our focus. Second, it's got to be affordable, right? We want to make solutions that are accessible to everyone, regardless of their background, their geographic conditions, their income. We want to make plastic replacements available to everyone. If it can be carbon negative, man, that's the dream. So I'm not making this claim that we have achieved carbon negativity already, but we're on that path. And there's a way to do that. As we know, seaweed has this wonderful sequestration potential. And at the end of its life, we also have the opportunity to emit fewer greenhouse gas emissions in landfill than other bioplastics and less than paper as well. And then on top of all that, it has to fit into existing infrastructure, and it needs to be high performance, it needs to do all the things that plastic does. And fortunately, seaweed is really well positioned to do that. So this is some of our material. This is majority seaweed content. You can see it's perfectly transparent, heat sealable. The shirt in there that I'm modeling this polybag with is from Pangaea, the company that utilizes seaweed in their clothes. So clearly there's an obsession here. Some of the other performance characteristics that we really like are that it has a 12-month shelf life. So you can think of it similarly to paper. If you're storing it in a dry condition on a pallet, it's gonna be fine. If you put it in a shipping container for 6 months overseas, that's hot and moist, it's not going to fare too well. So one of our biggest challenges is towing that line between maintaining rapid compostability and also, having a material that's going to last forever. And then some other benefits are that it is transparent, smooth, resilient. We can also deliver different types of features such as frosted films, different textures depending on the type of seaweed we work with or the amount that it's processed. You can work with semi-refined seaweeds as well. And central to all of this is, of course, the ocean. So I'm going to talk a little bit about our sourcing strategy and then I'll wrap things up. We're really focused on responsibly sourcing our seaweed. We need a lot of it to produce the amount of packaging we're focused on. So we primarily work with North and South American farms. The majority of those farms are farming and not wild harvesting, which is important to emphasize. We're interested in only adding nutrient content. And we try to adhere to the highest standard of responsible seaweed farming, the ASC, MSC standards, fair trade practices, as well as collaborating with ocean organizations such as the Safe Seaweed Coalition and local organizations such as Sonopexca and COPRAM to ensure that we're expanding the triple bottom line across the board. But we really, we understand that we may play a significant role in this emerging blue economy. So we want to really understand on a ground level what's happening in the water and what's happening for people. Erika, I hope this doesn't embarrass you, but we visited recently, and that's up in Humboldt. I was so happy. I got to hold my first nudibranch ever, and thanks to Erika here from Humboldt. We love to expand our relationships with the seaweed community, so we're so thankful to be here because we realize while we may slightly be outliers from the traditional attendee here. We really want to partake and make sure that we're not raising concerns about, overextending our presence in the water. There's my co-founder, Matt, down at Blue Evolution's farm in Erika, down in Baja. It was an awesome visit. We got to try Zertucci's wife's seaweed salsa. And then, of course, we'd love to be able to integrate some of these invasive species as well. So looking at what's happening in the Caribbean, working with sargassum, and then collaborating on the ground to expand regenerative scaling practices with these seaweed farmers to really improve working conditions across the board. We do have a little bit of traction, which is nice. We just won the Beyond the Bag Challenge, which was sponsored by CBS, Target, and Walmart. It's so nice to see the headlines, celebrating seaweed. But of course, we take it really seriously. These are billions and billions of bags that we're talking about replacing. So we need to be highly conscious of how we scale, and I welcome any questions or conversations from folks in this space. But I really would love for you to envision a world where we do succeed, which is imagine that every wrapper or bag or pouch that you interact with has actually encouraged ocean health. It's drawn down carbon emissions. It's helped to reverse the effects of ocean acidification. It's provided work opportunities for coastal communities. It's going to turn into healthy soil at the end of its life. That's such a wonderful potential that we have to actualize. And I have a lot of help to do it. Some of my team is here. This is us at the Botanical Garden, all waving in our nerdy little shirts, but we're really focused on impact. There's such a great collaborative opportunity here to alleviate millions and millions and millions of bags and wrappers and pouches, and to divert plastic waste from the ocean. So I hope that that wasn't too chaotic and resonated a little bit. Let's slay the future, guys. Thank you.
Presentation. I think we're all swayed. We're ready to go. Does anybody have any questions from the audience? Yes, back there.
That's such a good question. So we've done some initial testing with shoppers at Target to see what material manifestation is going to be most clearly compostable and seaweed derived. And what we learned is that while we do have the ability to deliver a fully transparent film that looks and feels just like plastic, shoppers are naturally drawn towards this sort of green textured film that immediately suggests biodegradability and that it is derived from a renewable resource. So we're going to continue to test textures, colors to see what people resonate with most. And you can see some of those samples over at our booth out on the lawn. I would love to get your feedback. But relatedly, we're also going through all the natural certification bodies, TUV, Austria, ASTM D6400, as well as BPI, to ensure that we're actually adhering to the standard of home compostability that we're claiming, and providing access through QR codes and smart labeling to information around local compost.
I guess kind of a follow-up to that question is like, half the time I go shopping, I go in like, well, shit, I don't want to buy that because it's just a plastic. So if it was more plastic, I would then purchase it. So how do I distinguish it at the time at the store? Like, can it work somehow? But when someone's in a hurry, I don't have to feel it.
Definitely, yeah. We want to work with brands who are excited to tell the story of seaweed, and they should be. I mean, it's incredible to invest in this new wave of seaforestation and ocean farming, so they should be proud to put it on their package right up front. They can center whole marketing campaigns around meet the farmer who grew your bag, So we see a lot of opportunity, and over time, we hope that the Sway name will equate to some level of trust and transparency. We're not afraid to admit the things that we're not perfect on, and you can visit our website and learn more about what our focus areas are to actually hit those benchmarks.
Thank you. Last one right here.
Yeah. So is the amount of seaweed that's being provided to the market limiting the growth of your company?
Not yet, not for the next 5 years or so, based on our current projections. We're working with a diverse range of farmers and a diverse range of locations, and we'll only continue to diversify because we never want to become too reliant on one species or one ecoregion. We do primarily focus on Mexico and Chile at the moment, but we'll continue to work with more and more farms. No, currently the bottleneck is the actual biorefining piece, not the seaweed piece. So yeah, also happy to continue talking afterwards. All right, over to you, Simona.
Awesome, thank you so much. everybody for sticking around. I know we're kind of running late, but I'm just going to do one last little talk. And so I'm coming to you from Ocean Era, so from the big island of Hawaii. And I'm going to tell you a little bit about your tax dollars at work, because we're funded through the Department of Energy, and we have some seaweed cultivation that we're doing. But before that, I just wanted to share kind of a little bit more of a personal story of how I got here, because I am one of the co-founders with Janet Kugler of this festival. And it's kind of amazing to be here and to see everybody here and to see the momentum and enthusiasm that people are having. And, you know, we want a more sustainable future. We want, you know, to address climate change, and we want to purchase products that are, you know, that are good for the environment. So, but I have to say, in 2002, I was at HSU, and I just started, you know, I started as an undergrad there, and I took a course called Women and the Environment, and it was just an elective. And I wrote this, you know, we had to, for our final project, we had to write a little, you know, report. And what I wanted to do, even back then, was I wanted to put a seaweed farm in Humboldt Bay. And like, that was, you know, that was my dream, but it was still, I think, so early, like nobody was doing that, you know, it was, you know, people were doing farming, but maybe, you know, land-based and doing different cultivation things there. And so I went on to get my master's at HSU, and I did a floristic study of Northern California, Southern Oregon, and just kind of found all the seaweed diversity that's up there. And it's incredible, the biodiversity that we have on this coast. And then I still wanted to do seaweed farming, and I was like, come on, does somebody, you know, want to like maybe guide me or something like that? And so I was introduced to Charlie Yarish, who's at the University of Connecticut. And, you know, the East Coast was kind of, you know, I wasn't sure about, you know, moving to Connecticut after living in beautiful Humboldt County. But I did end up going there and working closely with a lot of farmers in Maine. So my PhD was specifically on cultivating this and domesticating this very unique kelp species that they call skinny kelp, and is now considered a delicacy. But we were some of the first folks that actually domesticated it in the lab and then worked with farmers, put it out there. And some of the folks that really inspired me, like Sarah Redmond and John Kim, you know, Sarah was also involved in putting together a festival, because I think that it's so important, you know, to get the social license and to get people on board and excited, and, you know, and that's how you can get, you know, a lot of people don't want to see buoys in the water and things like this. And so if you share with people that there are all these ecosystem services and, you know, there are all these benefits, and we can do all these cool things, you know, with seaweed, with macroalgae, I think that's where things get to change. And so after Connecticut, I moved out to Hawaii, and now I'm working with tropical seaweed. So I'm going to share a little bit more about what we're doing at Ocean Era. And so again, this is funded by ARPA-E. So we've got a nice grant. Yep.
5 minutes.
5 minutes. All right, I'll go just. Global seaweed production, it's all based in Asia. So this is why we got to change this. Offshore seaweed aquaculture has so many benefits, right? So, you know, carbon removal, bioenergy production, feedstuff, fuel, feed, food, fish habitat, all these cool things. This is the facility at Ocean Era. So we have a lot of land-based tanks, and we're investigating some different species that we can cultivate that could potentially go offshore. So I think for scalability purposes, we do have to think about, you know, farming the ocean. And so we have to do some trials on that. So we're currently testing some different species, several sargassum species, Ulva, Lactuca, Halimenia, Hawaiiana, Gracilaria parvispora, and Caulerpa lentulifera. And these are all native Hawaiian species that we're working with. You know, and just the trials and tribulations of growing seaweed. So, for example, with this Ulva lactuca, it kind of dissociates at different times, but we're trying to use that to our advantage and seed little spools so that then we can take the seed string and put it onto lines. Okay, so here's some little Ulva propagules that are 2 to 5 millimeters. And then in a few months, we get to see, you know, almost a couple kilos, a kilo of growth. With Halimenia, we've mostly been working with vegetative, with just vegetative propagation, but we're also working with some of the spores and trying to figure out how to get those onto lines. Because again, we do see good growth with Halimenia, but you know, if you take that offshore, you want to make sure that your lines, that your seaweed is attached to your lines. With sargassum, it is problematic, you know, in the Caribbean, but if we're using the native species, there is some potential, and we're working with different ways to go through the life cycle, kind of filter at different levels, concentrate those, and also put the little zygotes onto spools. The only thing is we've been having some trouble because they grow very slowly. So we'll see how that one goes. Caulerpa lentulifera, this is sea grapes or vegan caviar. So this is a delicacy in the Pacific Islands. And so we're looking to see how we can grow that efficiently. And then this one, Gracilaria parvispora, we're working through the life cycle to try to isolate the carpospores and grow clean individuals. So strain selection is also super critical. And, you know, just different, I'm not going to go through this, but just, you know, different experiments and things like that are super important. And then finally, I just want to mention this. So we're putting an array offshore off of Kailua Kona. And, you know, it's going to be unidirectional with the current. And so we're going to see if, you know, the current will provide enough nutrients, because these areas are very low in nutrients. So they're oligotrophic, but we'll see if, you know, the nutrients will basically allow for the seaweed to grow in this environment. And we're also working with Mackay Ocean Engineers on that, and also a great team at UCI and also at University of Hilo. And this is also really important, kind of thinking about the life cycle analysis of the farm, thinking about carbon moving through the farm. And this is obviously super critical for thinking about carbon sequestration. So there's my talk in a nutshell. Thank you so much.